Bismillah ar-Rahman ar-Rahim. This is the second video on the June 21 paper 4.1, and this we will be handling the questions after the question 4 and 5. Starting with question 4, genetic engineering involves the manipulation of naturally occurring enzymes and processes. List the enzymes used in genetic engineering and outline their roles in natural processes. So, which enzymes have we used? And then what were their roles in the natural processes? But of course, we are now using them for genetic engineering, but we figured them out because they were actually being occurring in living organisms. We found them. We didn't invent them. We discovered them. Now, as you can see here, I've grouped it into 4 different categories. The first one is I've used 4 different colors to make it easier for you. The first one I've talked about is restriction enzyme. So restriction enzyme cuts DNA. Where was it found? We found it in bacteria because bacteria has its sort of an immune system by which it can cut and recognize alien DNA. So there are certain bacteria which are attacked by viruses, and when the virus DNA is injected, these bacteria can then cut up this DNA. So the bacteria actually knows, and these viruses are called bacteriophage, and the phages are the bacteria cuts up this alien DNA. So we discovered these. I said discovered, not invented. Discovered these restriction enzymes from bacteria, and that's why they're named after the bacteria, like EcoR1, E. coli restriction enzyme 1. So restriction enzymes cut DNA produced by bacteria to cut viral DNA. So it is a protective mechanism, just like we have lymphocytes which make antibodies. Then the other one which I've talked about is DNA ligase, which joins DNA like we use it in joining the Okazaki fragments, which is the lagging strand, the leading and the lagging strand, which we studied in the AS level biology. Then reverse transcriptase, where did we find it? We found it in retroviruses. In retroviruses, R-retro, rRNA. So when the RNA has its own reverse transcriptase, when it injects this RNA into a host cell, then from that RNA, the viral DNA is made. So reverse transcriptase makes cDNA from, but actually we use it to make cDNA from the mRNA. Like for instance, I want to make insulin, so I will take the mRNA from the pancreas, beta cells of the pancreas, and then give reverse transcriptase and make cDNA out of it. And is present in retrovirus. And then the fourth one is DNA or Taq polymerase, which actually just does a copies DNA replication. We use it in PCR, polymerase chain reaction, and it is present in the thermophilic bacterium called Thermus aquaticus, which can of course survive very high temperatures and found in hot springs. So 5 marks, and you have to give me any 5 out of all these. A whole lot of marking scheme, nearly 12 mark scheme points. Now coming to part number B of this. Genetically modified organisms, GMOs, include crop plants. Genetically modified crop plants have been grown in North America in increasing quantities since 1996. They are now grown in many areas of the world and are eaten by millions of people and farm animals. Table 4.1 compares the area of land used to grow GM crop plants on 6 continents in 2013. Now, we're looking at a whole lot of continents: Africa, Asia, Australasia, Europe, North America, South America. Area of land used to grow GM crop plants in million hectares. So 3.5 million, 19.1 million, 0.6 million, 0.2 million, 81.1 million, and 70.9 million. But we don't know how big these continents are. Is it a certain percentage of their total area? We don't have no idea. These are the things which you need to think. We just know, okay, this is the million hectares. In Africa, 3.5 million hectares has GM crops. In Asia, 19.1 hectare. And Asia is a continent. It's not, we don't even know which countries or whatever. We have no information on that. Then the edible GM crop plants grown. So these are the edible ones: maize, soya bean, maize, papaya, tomato, canola, maize, papaya, and squash, sugar beet, and canola, maize, and soya bean. So you read this table and then you have some sort of a rough idea, okay, what we are talking about. Uh, Part 1 says suggest how North America and South America benefit from growing large areas of GM crop plants rather than non-GM crop plants. Well, of course, because there's a higher yield. GM crops we've genetically engineered them, so they give us more yield or more food. So increased higher yield, higher quality of the crops or the food. We use less of the insecticide and the pesticide, and we lose less fuel. And then we set aside conservation areas because less is needed for crops because the crops are high yielding. And comparative economic benefit, you see, the non-GM crops cost the same, but they give less yield. So economically, the GM crops are much more fruitful. Just the wordings have given you higher yield, better quality of food, less insecticide, less fuel, yes, CO2 emissions, set aside conservation areas because now we will need less area for the food crops, and economically it will be beneficial because it will be higher yielding than the same amount of land used, but the same area of land used, but more crop will be harvested. Part 2, only a small area of land is used to grow GM crop plants in Europe. So if Europe is 100, we're only using 2. I mean, I'm just giving you an area of only a small area of land. This is mainly because most of the Europe uses a precautionary principle. The precautionary principle prevents GM crop plants being grown if there is a possible risk of harm to human health or to the environment, even if there is no proof of harm. So they are very extra careful. Many European citizens have concerns about the safety of eating GM food, but Europe imports large quantities of GM maize and GM soya bean to feed farm animals. The farm animals are ultimately going to be eaten by humans. So Europe is importing large quantities of GM maize and GM soya bean to feed farm animals. Use this information and table 4.1 to deduce two arguments in favor of growing more GM crop plants in Europe. Because you see, people in other continents eat GM food, and GM food is already in the European food chain. Why? Because it's being fed to animals, and then those animals are being slaughtered and by eating humans. And economic benefits of reducing GM animal food imports. So all this is an economic benefit. So it was in reduce these arguments in favor of growing more GM crops. So people in other continents eat GM food. Why is people in Europe not eating them? Then GM food already was in the food chain because the farm animals were eating it. And then the economic benefit because and we will reduce the import because we were importing these. We were importing large quantities of GM maize. So if we grew it in our own country, then we won't need to import it. So if Europeans grew it in their own country, then they won't import it, and then they would just have their own. They won't extra money won't be spent on the import. Then coming to the last part of the question, part 3, explain why the data in table 4.1 are not enough to calculate the extent to which different continents have replaced traditional crop plants with GM versions. So this data here, why is this not good enough? Why? Because we do not know what is the total crop area or what is the non-GM crop area. If this was 3.5 million hectares, how much was the non-GM crop? Was it 10 million or 20 million or only 1 million? So we do not know the total crop area or the non-GM crop area. So we cannot work out the percentage of GM to non-GM. We only know that 3.5 is the GM crop area. We only know 3.5 million hectares is the GM, but we don't know how much is the, so we cannot work out the fraction that, okay, so 20% of it is GM crops or 30% or 40% is. So we have no clue how this is. So this was a question which was addressing this part. Explain why the data in table 4.1 is not enough to calculate the extent to which different continents have replaced traditional crop plants with GM versions. And this was 2 marks. So just a very easy way to word it, need to know total crop area, 1 mark. So can work out percentage of GM or non-GM crops. Now coming to question number 5a. Nerves consist of bundles of different types of neurons. Figure 5.1 shows the mean transmission speed of impulses for three types of sensory neurons, C, A delta and A beta. So A delta and A beta, now you can see here what do we have? We have the mean axon diameter. So the mean axon diameter 1, 3 and 9, it's in micrometers. So 1 micrometer mean transmission speed of impulse was C was very little. It's 5, 1, 2 only. Then 3 and then 9. So as the axon diameter is increasing, the mean transmission speed of the impulse is also increasing. 3 was nearly 18, and the one which was A delta and A beta was 9 micrometer in diameter, and it was 55. So you can see here, I mean, I'm trying to get this right here, 55. So this is what I'm looking at. Then I'm looking at this one, and this is now 18. And here, this is only 2 here. So let's look at it very carefully. Mean transmission speed of impulse is in milliseconds. And this is 2, and then in one it is 18, and one it is 55. Let's look at the question now. Impulses from pain receptors in the skin are sent along sensory neurons C and A delta. Sensory neurons C and A delta synapse in the spinal cord with relay neurons known as projection neurons. Projection neurons send impulses to the part of the brain that perceives pain. Impulses from touch receptors in the skin pass along the sensory neurons A beta, which can also synapse with the projection neurons in the spinal cord. Sensory neurons A beta and A delta are myelinated. Sensory neurons C are unmyelinated. So a lot of information, you need to read this again, you need to understand it, that how they are trying to show you something. So pain receptors in the skin sent along C and this, A delta. Sensory neurons C and A delta then synapse with relay neurons. Then these relay neurons send impulses to the brain to perceive pain. That's why we feel pain when a pin hits us in the foot or something. Impulses from touch receptors in the skin along sensory neurons A beta, which can also synapse with the project. So from the skin also we have these neurons going. Then sensory neurons A delta and A beta are myelinated, while the C are unmyelinated. So pain receptors of the skin are C. So pain receptors in the skin are C, and these are unmyelinated. And sensory neurons C synapse in the spinal cord with the relay neuron. Okay. Now describe the results shown in figure 5 and suggest explanations for these results. Wider, thicker, larger diameter increases speed. Yeah, that's what we figured out from here. That the wider they are, the thicker they are, or the larger they are in diameter, they are higher the speed. Then the larger membrane surface area, so more ion movement. Then myelin increases the speed. Myelin sheath, increases the speed. Then saltatory conduction. Saltatory conduction is when it, of course, jumps from node to node. And then you could give me some data comparison. You could give me, okay, in 1 micrometer diameter, 2 milliseconds, 2 micrometer per millisecond is the rate, while in 9 micrometer it is 55. Speed is 55 per millisecond. So you give me a data code and you got that. So you have to understand that whenever it says describe the results and suggest explanation, so that means you have to give me a describe as well as an explanation, because there are 3 marks for this. So larger diameter, higher speed, fine. More surface area, so more ion movement. Then myelin can increase the speed, because it said suggest explanations as well. And saltatory conduction in the myelinated ones. And then of course you gave me the data from it. Then gently rubbing a damaged area of the skin can reduce Gently rubbing a damaged area of the skin can reduce the perception of pain. Suggest an explanation in this. Now, you see, you have to look at it. Everything is coming from the question. It's nothing information which you need to know from the book. It's all question. It's all the information is given in the question. Now, part two, gently rubbing a damaged area of the skin can reduce the perception of pain. Suggest an explanation for this. Now, you have to see it was all in the question. The touch receptors were here in A beta, and A beta travels the fastest. This is one A beta. This is the one A beta. So the answer was correct. The impulses from touch in A beta neurons travel faster. So this everything was from the question, nothing from prior knowledge. And this will inhibit the relay or the projection neuron, so the person is not going to feel pain because it said reduces the percentage of pain. So if you know how to answer these questions, that's a very big art of answering this question, is that you find out the answers from the question itself. It said gently rubbing, so this is a touch sensation. Then reduce the perception of pain. So it will inhibit those relay neurons which were going to transfer this. The brain was going to be informed and you were going to feel the pain. Right? So please remember, read the question carefully, understand the question carefully, take your time, and then answer it in a very befitting manner. Now, coming to the B part of the question, back pain in humans can be reduced by using a transcutaneous electrical nerve stimulation TENS machine. A TENS machine uses electrical impulses to stimulate nerve endings near the site of the pain. It is thought that TENS triggers the release of natural painkillers called endorphins at synapses. Figure 5.2 shows a synapse in a pain pathway. So look at it very carefully. Impulses from area of painful back muscles, then endorphin receptors, then presynaptic knob, then postsynaptic neuron to the pain center in the brain. Here you have acetylcholine receptors. Here you have vesicles containing acetylcholine. Here you have vesicles containing endorphins, and impulse from area stimulated by TENS machine. So they've given you a new situation to understand and comprehend, but you have to remember your synapse story so that you can then correlate it with this, right? Now, suggest and explain how endorphins may act to reduce pain. So how are endorphins going to work? Very basically, endorphins will bind to the receptors. They will bind to the endorphin receptors. They will stop the calcium ions from entering the presynaptic knob. So lesser vesicles or no vesicles will move towards the presynaptic membrane. So no acetylcholine released, no binding of acetylcholine to postsynaptic receptors. So no depolarization of postsynaptic neuron. So action potentials, so there'll be no action potentials or impulses in the pain center or the brain. So all that which was happening in a normal synapse is not going to happen. Recap it on this diagram. So these are the vesicles containing endorphins. So endorphins will bind to receptors. So this endorphins will bind here to the endorphin receptors because here we have the endorphin receptors. So if the endorphins will bind to the endorphin receptors, this will not allow calcium ions to enter. So calcium ions cannot enter anymore. Calcium ions cannot enter. Stops calcium ions entering the presynaptic knob. Please remember, this is the presynaptic knob, this is the postsynaptic neuron, this is the presynaptic. So calcium ions will not enter. So now vesicles will not move. So these vesicles, let me give them another color. These vesicles will not move and will not fuse with the presynaptic membrane. This is the presynaptic membrane. So either no, or you can say fewer vesicles will move towards. So less acetylcholine will be released. So maybe there's just a little acetylcholine will be released. So less acetylcholine released, less binding to the postsynaptic neuron, or no binding to the postsynaptic receptors, and so there will be less depolarization. So action potentials in the postsynaptic neuron will be lesser. or you can say no depolarization of the postsynaptic neuron. So less action potentials will impulses to the pain center in the brain. So there will be less action potentials to the pain center in the brain. So the person is not going to feel the pain. Then coming to the last part of this question, suggest one disadvantage of using pharmaceutical drugs for reducing pain compared to using TENS. Yeah, well, drug dependency or addiction or tolerance. I mean, you take one brufen, now you have to take two brufens, and brufen has a lot of side effects, and that results in addiction or dependency. Then side effects. I know I have a knee arthritis and I have to take brufen, and it has very bad side effects on my stomach. Then it's expensive, and then work very slowly. They work very slowly. It takes a lot of time before it starts to have its effect on you. You might have swallowed it about half an hour ago, but it still doesn't start to work. So there was only one mark, but I mean, these are the sort of answers you could have given. Of course, against drugs is, drug dependency, side effects, expensive, cost money, and work slowly. Now, question 6, thermoregulation is the control of the core temperature. Now, this is no longer in the present syllabus, so I will not discuss this question because this has been removed from the present syllabus. So, thermoregulation is no longer in the syllabus. I'll Now we're left with question 7 and question 8, which we will do in the next video. So, thank you very much for watching, and wish you all the best.